Hello, this is Miss Peachy from WCA Biology A, and I'm doing this just overview of Unit 2, Lesson 6 on mutations in biology. So I'm starting here at slide number 3, just because the first two slides kind of went through an overview of stuff we've already learned. One of the concepts, that was mentioned in the first two slides that really wasn't talked about previously is kind of this idea of gene expression and how um, not every gene that you have is being expressed at all times. So you have in your cells, in your nucleus of your cells, your DNA, you have a complete copy of your DNA in every cell of your body, every place in the whole body. But you don't have to express all of the information that's coded in that DNA in every place all the time. And I think that's pretty obvious, that certain cells specialize and code for specific things, right? For example, one of the things I talked about was melanin, which is found in human skin. And melanin produces a dark pigment in human skin. And many of us have more or less, kind of depending on the pigment. Some of us have a little flecks of melanin. If you've got freckles or if you're dark skinned, you have more melanin. But that melanin that is produced protects us from ultraviolet radiation, and it's necessary in your skin to help protect from skin cancer. However, you wouldn't need, for example, to have the gene that codes for the production of stomach acid to be expressed in your skin. It wouldn't make any sense. Then your skin would produce stomach acid. You're not digesting food in your skin, so we don't need to do that, right? So that's regulation of gene expression. So you're not expressing every gene everywhere all the time, even though every nucleus of every cell contains a complete copy of all the directions. So sometimes there's a problem in how those proteins are coded for. So the DNA is made up of these nitrogenous bases, A, T's, C's, and G's, and every it's transcribed into messenger RNA and then it's translated into amino acids. But if there's a problem with the code, then the process of transcription and translation can be affected as well. And that's what a mutation is. So before we kind of move into the content of the lesson, I want to pull something up and show you. And I didn't mention this in our first two videos because I kind of just started making these. This is all kind of new this year. I'm going to bring this over here. On my website, you will find this document. So it'll be linked there on our biology website. And this is basically a notes guide or a graphic organizer for this lesson. So as you're completing and watching the video or going through the lesson, you can write your information down in this notes guide, in this graphic organizer, and it will kind of follow the order of the lesson that I'm going to be presenting to you now. So just kind of have that up in front of you. You can do a split screen or something and kind of type as we go, but that's going to help you out here a little bit. So let me bring the lesson back up here. Okay, so the definition here. The definition is of, let me just get mine actually up here to make sure that I'm following along as well. So the definition of a mutation is a change in the genetic information that can affect the production of proteins and in turn gene expression. And what can cause mutations? They can occur because of natural events, environmental conditions, or even by artificial means. So perhaps, you have a lot of solar radiation can cause skin cancer, and that's a mutation. That's also kind of an environmental condition as well, but it can lead to environmental conditions, maybe excess amounts of lead in your water or something like that. Those are all external environmental conditions, or even by artificial means, like, for example, if you receive treatment for cancer, some chemotherapies or some radiation treatments for cancer can actually also cause mutations as well. So many mutations have little to no effect on gene expression, but some can be harmful. So some are neutral, which means they have very little to no effect. Some can be harmful. For example, they talk about something called cystic fibrosis, which is an inherited disorder, and it says it is caused by the mutation in which 3 bases have been deleted from a specific gene, the CFTR gene. So these 3 bases that have been deleted cause a mutation. This is a bad mutation that basically causes this protein that's in the membrane surrounding every cell is unable to deliver chloride ions into the cell. So you can't get chloride ions into the cell, and that results in this thick mucus that can build up. It builds up in the lungs, which makes it difficult to breathe, and it also can build up in the intestines, which actually reduces the absorption, prevents the release of enzymes from the pancreas into the intestines, which then also reduces the absorption of nutrients. So you become malnourished and you can have growth issues and stuff like that, all because of this 3 different bases that have been deleted, right? So that's a bad effect. It can also cause a helpful effect. For example, many people do not have the enzyme to help digest the enzyme or the sugar lactose. So lactose is a sugar that's found in milk. And if you want to drink milk or eat milk products and you don't have this enzyme that helps you break down sugar of lactose and get the nutrients out of it, you can actually feel pretty sick if you don't. But some people have a mutation that allows them to do that. And so that helps them be able to extract nutrients from foods that they may not otherwise eat, like milk from goats and cows. So if you have the mutation that allows you to digest lactose, that's actually a mutation, then you can actually eat milk products and drink milk without any problems, and you can get more nutrients from them. So that's a good way, a good effect of a mutation. So we have different kinds of mutations. Most mutations are single point mutations, and we're going to talk about point mutations because there's a couple different kinds. You can have substitution mutations, or you can have frame shift mutations. Those are both called types of point mutations. So a point mutation is a change in a single base, one single base, and how it's going to kind of lead to other effects. So one type of a point mutation is called a substitution mutation. A substitution mutation is when one base is kind of removed and another base replaces it. It's substituting it, right? So it only affects one base, and therefore it only affects one amino acid in the entire protein. There are 3 types of substitution mutation kind of effects. One is a silent mutation, one is a missense mutation, and one is a nonsense mutation. For a silent mutation, and it helps if I bring this up here, what happens is one of the bases, in this case G, is deleted and substituted, A is substituted. So this alters our sequence of DNA. It was TAC, AAG, ATG, now it's TAC, AAG, ATA. Transcription, that's changed into messenger RNA, so that is also altered. This codon was UAC and now it is UAU, but thankfully in this particular situation, both UAC and UAU both code for 309 here. So they both code for the same amino acid, so yay! There is no change in the amino acid sequence, so it's known as a silent mutation because even though there's a mutation, it's still coded for the same amino acid, so the same amino acid sequence is produced. So you won't even know if you had this. Then we have missense mutation. And a missense mutation, again, is where you have one of the bases that's plucked out and a new one is substituted. So in this case, T is removed and, in this case, it should be A. A is removed and T is substituted. That changes, again, the messenger RNA. But in this situation, it actually leads to a change in the amino acid. Okay? So you have a different amino acid here where you didn't have that before. So this could be bad. It could really make the whole thing not work at all. Like if you take one amino acid out and put a new one in, maybe the whole protein doesn't work. Or it could be kind of okay because some amino acids are very, very similar to others. So sometimes you can substitute one and it isn't a big deal. It can really not lead to a huge change in that protein and it still functions okay. So it just kind of depends on, how close, I guess, the substituted amino acid is to the original. If it's really very similar, then it's probably fine. If it's very different, then it probably be a problem. And then we have our nonsense mutations. And so this happens again when you have a substitution. So G used to be G and C is substituted. And what happens when you go through the process of transcription and then translation is that this used to be an amino acid. I guess this is, I think it's actually a tryptophan, so I think I misspoke earlier. But it used to be an amino acid, and now it has been substituted with a stop codon. So basically that stops translation at that point. And that's a problem because that means that your whole protein isn't going to get made. So if that stop codon occurs kind of towards the beginning of that whole chain of amino acids, it's going to really mess everything up. And then you will not be able to have a complete protein. So it becomes kind of a big problem. If it occurs towards the end, then the protein just gets shortened a little bit and maybe it's not as big of a deal. But it really depends more so on where it occurs, where that stop codon occurs. So those are our substitution mutations. Then we have our frame shift mutations, and these are also point mutations as well. So with our frame shift mutations, we have deletions and insertions. So instead of taking one nitrogenous base out and substituting another one, you're actually removing a whole nitrogenous base and just nothing replaces it, or you're adding an extra one. And what happens here is something called a frame shift. Because if you can see here, and this is before mutations, remember that your whole sequence of messenger RNA is read 3 bases at a time. And every 3 bases is a codon. So AUG, that's one codon, and that's coding for this amino acid, right? And CGU codes for this amino acid. So if we add an extra one, the first amino acid still codes correctly, but now every other one is going to be wrong because you added something extra. So every 3 is different from what it was before. So the entire chain is incorrect. Same thing occurs with deletion. Now you're deleting one out of there. So the first one codes correctly, but now every single one after the deletion is going to be wrong. So it depends again when the insertion and when the deletion takes place. If this happens towards the very beginning of the chain of amino acids, it's going to affect the entire thing and it's a really bad thing. If it happens at the very end, it may only affect a few different amino acids. So again, it depends kind of where it occurs, but it's going to affect multiple amino acids. So oftentimes it's a much more serious mutation than a substitution would be. Finally, we have our chromosomal mutations. And these occur when the whole chromosome itself, the structure of the chromosome, kind of gets messed up. What can happen is during cell division, when a cell makes a copy of itself, it has to also make copies of its chromosomes. When that happens, sometimes you can have part of the chromosome that actually gets broken off and doesn't get replaced, so it's lost, or it can kind of get stuck to another chromosome, so you get 2 chromosomes that combined information from one, or maybe it's duplicated, so there's more than one copy of the chromosome that used to be there. So all of these can lead to problems in the cells and the proteins that are made from these cells. So that's called a chromosomal. And these are the 4 different kinds: inversion, translocation, deletion, or duplication. So inversion is when the chromosome kind of gets flipped upside down. Translocation is when you have chunks that get kind of broken off of one and pasted onto a different chromosome. So they're kind of like mixed up. Deletion is a whole chunk gets left out. And duplication is where like a chunk of the chromosome gets copied twice. And so now you have 2 chunks, where once there was one. So it might code for more proteins to be made because there's 2 copies of it. So those are our types of chromosomal mutations that occur. And since they're parts of the chromosome, then they're going to affect every other cell that eventually is copied from that particular cell. All right, so that's an overview of Unit 2, Lesson 6, Gene Expression. It's the end of this segment, and bye for now, and I'll see you in the next one.